and welcome back to this module 6, the last session of protein and protein based products. So, I will proceed with this last session, and I would thank all of you to be patient and patiently listening to me throughout. And also I am deeply appreciating the efforts that have been put by various people, from where on the website I have actually accumulated the information, and I acknowledge them from the bottom of my heart. I again re-emphasize that all these informations have been only used for the purpose of teaching and learning. No infringement or copyright violation is intended in these presentations. So, I will proceed with the last module, that is protein based products. So, from the commercial point of view, in the last class I told you about the various ways how commercially viable protein products are made in huge quantities. So, from the commercial point of view, the proteins may be classified into the following categories. Number 1, blood products and vaccines. So, for example, there is a factor called factor 8, and then there is another factor called factor 9. Both of these factors are protein products which are obtained from the blood, and they are used typically in hemophilia, which is a hemophilia A, which is a classic hemophilia, or hemophilia B, which is called the Christmas disease. So, these two are hemophilia. Hemophilia means, there is when the blood starts coming out from any wound or any surface from inside, within the body, etc., it would simply not clot. Ideally, what happens is, if you get a cut mark and the blood comes out, then after some time it just the blood clots and the blood stops to flow. But hemophilia is a situation where the blood will keep on oozing out. So, the blood clotting factors does not work. So, hemophilia A is caused by lack or decrease of the clotting factor called 8, and hemophilia B is caused by the lack or decrease of the clotting factor called 9. So, these two proteinaceous products are now being synthesized by various kinds of engineering technologies, in which the bacteria has been tweaked to produce these factors, and these factors are nowadays utilized. So, they can be produced in large quantities from a commercial point of view. Another important one is the hepatitis B, and this is one kind of a hepatitis infection. Hepatitis B is a potentially life threatening viral liver infection caused by hepatitis B virus, and the vaccine for the prevention of this hepatitis is now being synthetically produced, and they have been produced in commercial point of view, that is in huge quantity. So, another important protein product, which is when we use the genetic material to clone a particular protein for vaccine production. So, one example is given here. So, you see hepatitis B, which is a potentially life threatening liver infection causing virus. It is caused by the hepatitis B virus, and so if you wish to get vaccinated against this virus, you need to prepare those vaccines, and for that what people do is, they have taken this virus particle, and then the genetic material of this virus particle, the section of the genetic material which probably codes for surface antigens or is responsible for immune response from this in the human cells, has been taken, and that particular portion of DNA is now put into this plasmid or a vector, and that is now injected or transformed into the yeast cells, or even in some other cases some bacterial cells, and then these yeast cells can be grown in culture, and large number of the genetic material will be translated to produce the proteins or the surface antigens called the HBS, or hepatitis B surface antigen, and that antigen can be used after purification as vaccines. The second type of product is therapeutic antibodies and enzymes. So, these are huge markets all around the world. So, polyclonal antibodies have been used for more than a century for therapeutic purposes, whereas more recently monoclonal antibody preparation as well as antibody fragments are produced by recombinant DNA technology and have been found therapeutic use. So, what is the difference between polyclonal and monoclonal antibody? Polyclonal antibody, as you can see, these black portions are the polyclonal antibodies, and if you see, they have the surface binding ability to bind to any bacteria or virus or any other pathogen or any other foreign material. Now, this foreign material on its surface would have large number of antigens or so called stimulating agents, which is recognized as foreign by our body, and therefore, our body produces a huge amount of antibodies, each color representing specificity towards a different antigen on the surface of the foreign body. So, if you raise a large number of antibodies which is non-specific to any particular thing, but actually towards the entire let us say antigen, allergen, bacteria, virus or whatever you talk about, then those are called the polyclonal antibodies. So, they are cheap to produce because you just have to produce those antibodies by injecting the foreign body inside a body, and then you get a mixed population of antibodies. This antibody, for example, is specific for the red one, this one is specific for the green one and blue one and so on, and they may bind to different areas of the target molecule. Here let us say the target molecule is just a bacteria and it is tolerant to small changes in the protein structure. So, these are the polyclonal antibodies. If you look at monoclonal antibodies, so this was used, this particular process was in use long back and has been used even nowadays. So, you just have any virus or bacteria and inject it into human or another animal, and after some time you bring out the serum, and the serum will consist of antibodies against that bacterium. However, if you want to have a monoclonal antibody, they are very specific and they bind to only one particular antigen. So, they are expensive to produce because you are trying to be very specific to one antigen, and then there is a single antibody species will only bind to specific site and may recognize a particular protein from the monoclonal antibody. So, this picture has been taken from lab clinics with the website given here. So, this is the difference between polyclonal and monoclonal antibody. The monoclonal antibodies are now typically generated or produced by recombinant DNA technology, where this particular portion of the antibody is infused into a bacterial cell, and the bacterial cell is now tweaked to produce that antibody of interest. There is another another monoclonal antibody called the OKT3 here is used to prevent rejection following kidney transplantation, because the antibody blocks those immune cells which attack foreign grafts. So, when you have for example, a diseased kidney, and then this person receives a good kidney from a donor person, and then that is transplanted, many a times what happens is, because for this person the donor kidney is a foreign material, therefore, this person immediately generates antibodies against this particular kidney, and therefore, many a times it gets rejected and further generates various complications. So, what people have done is, now they have got an antibody, monoclonal antibody of named OKT3, and then if you inject this recipient along with that OKT3 antibody, then the rejection does not happen, because that antibody will neutralize the normal antibodies against this foreign kidney. So, that is kind of a very useful technology, and with that people have been able to have this kidney transplantation much more effectively nowadays without the fear of rejection. Fragmented portions of therapeutic enzymes, for example, fragmented therapeutic proteins, for example, tissue plasminogen activator. This is a proteolytic enzyme and it is a serine protease. If you remember in my previous discussion in the previous class, I have told you how a serine protease works. So, there is a catalytic triad. So, this tissue plasminogen activator is another serine protease, and it is used to digest block in arteries. So, this tissue plasminogen activator actually converts plasminogen to plasmin, and once this gets converted into plasmin, if there is a blood clot somewhere, this plasmin will just break that, and therefore, the arteries will be cleared and blood flow happens. So, myocardial infarction, that is heart attack in simplistic terms, because of blockage of arteries can easily be taken care of by this recombinant tissue plasminogen activator. So, these enzymes or these proteins are now being synthesized and they have been of immense therapeutic purposes in large laboratories in large scale. Proceeding towards the third type, therapeutic hormones and growth factors. So, a number of hormone preparations have been used clinically for many decades. I would like to draw your attention here, insulin. And many of you might have at your home elderly people who are suffering from what is called as diabetes. So, what happens is the sugar level in the blood increases and then they require some injection called the insulin injection to be taken, or insulin tablets, or whatever different types of medications to reduce the blood glucose level. The very potent and immediate effective function is the use of insulin, which is a hormone. Now, this insulin was so far or originally being produced from the pancreas of cows and pigs. So, the cows and pigs were slaughtered, their pancreas were taken, minced, and this particular insulin protein was isolated or purified, and that insulin have been used so far till few years back, have been used extensively for people who are suffering from diabetes and requires insulin. But now you cannot keep on doing that, because the total population of people suffering from insulin dependent diabetes is just increasing, and it is very shocking that it has been predicted that India is going to be the diabetic hub of the world, with 20 million people infected or affected by this diabetes problem. So, we need huge amount of insulin. Where to get? We cannot just keep on slaughtering these cows and pigs, and therefore, this protein or this hormone insulin has been genetically synthesized from the human cells itself. So, what has been done is you take a you take a bacterial plasmid, and to that bacterial plasmid you attach, by using some enzymes called the restriction enzymes, you attach a gene responsible for producing human insulin, which is shown in red here. And then this entire plasmid is reinserted into the bacterium, and the bacterium is allowed to grow by the fermentation technology that we have learnt earlier, and from there by different purification procedures we can get the insulin ready for distribution. This insulin is now, because it is obtained from human gene, it is called humulin, therefore, it is humanized insulin, and humanized insulin has been found to act in 15 minutes, whereas those which were obtained from cow and pig used to take 3 hours. So, this is one success story which has been now very commonly used. Another one is platelet derived growth factor, or PDGF. It has been approved for diabetes who develop skin ulcers. So, this is another PDGF is also now synthetically produced or engineered into bacterium, and we can actually get these growth factors, and therefore, can be used therapeutically. Several other growth factors, for example, interleukins etc., are under various stages of clinical trial, which I will be talking to you in a short while from now. Moving on, other industrially important proteins are regulatory factors. Several new regulatory factors were discovered that did not fit the classical definition of hormone, and initially they are known as cytokines. So, these include, as I was just now telling, the interferons, interleukins, IFNs, interleukins, ILs, tumor necrosis factor TNF, and colony stimulating factor, or CSF. So, for example, IFN alpha or interferon alpha is used for hepatitis C treatment, interferon beta is used for multiple sclerosis treatment, and interferon gamma for chronic granulomatous disease. So, this chronic granulomatous disease is a disorder that causes immune system to malfunction. So, when a foreign antibody enters, ideally you should develop an immune response. But if you have the chronic granulomatous disease, then the immune system doesn't respond and they malfunction, resulting in form of immunodeficiency, and then the person suffering from chronic granulomatous disease will undergo multiple infection from various kinds of viruses and bacteria. The other one, the multiple sclerosis, is a problem which is more to do with the nerves. So, what happens is the nerve cells are generally, the axons of the nerve cells are generally covered by a myelin sheath, and this helps in the movement of nerve impulses from one end to the other. That means when you are blinking your eyes, your hands and legs are moving as you think. So, those motor activities are done by transmission of information from one nerve to the other through these axons, and there are these sheaths on top of it which helps in movement of the information quickly from one end to the other. But then in multiple sclerosis, what happens is these myelin sheath gets demyelinated, and once they demyelinate, there is leakage of information all along the axon, and that causes various neuronal problems. So, that can be corrected by the presence of interferon beta. So, interferons have such large effectiveness, and they are now being produced classically by recombinant DNA technology in huge quantities. Moving on, the proteins can be used in analytical applications. For example, enzymes and antibodies have found a range of analytical applications in the diagnosis of diseases. For example, hexokinase is used for quantitative estimation of glucose in the serum. The uricase is routinely used for measuring uric acid levels in the serum. Two very important enzymes, horseradish peroxidase and alkaline phosphatases, are used in ELISA. And then there are hundreds of applications that we generally do every day in our laboratories. For example, the recombinant DNA technology work, which requires large amount of restriction enzymes, the polymerases, the PCR machines, you might have learnt about them in your previous classes in some other context. So, all of those enzymes, the lipases, the ligases, the restriction enzyme, the topoisomerases, etc., all these enzymes are used in analytical applications or in the laboratory scale applications for doing various kinds of genetic engineering processes. So, all of those are being synthesized in industrial scale nowadays by methods of genetic engineering. In the industries, there are many enzymes which are used. One particular enzyme called the proteolytic enzyme, which includes a large set of enzymes, not only the serine proteases, there may be aspartate proteases, there may be other proteases. So, proteolytic enzymes, also called proteases, constitute an 8,000 crore annual market. Look at that for the enzyme, for the industrial enzyme, and they find in applications in, let us say, beverage industry. For example, the papain, it is used in all kinds of beverage production. They are used in detergent industry. For example, another protein called alkylase, you see these are enzymes. So, they are used in detergent industries. Then you have the confectionery industries. So, there is an enzyme called glucose isomerase, which is produced in huge quantities and is responsible for production of bread and other things, other confectioneries, bakeries. You have cheese production. So, there you need to have this enzyme called chymosin, which is again important for cheese production. And then leather processing and meat industry, you have huge quantities of lipase requirement. So, with that, we now know what are the different aspects, how broad is the spectrum of the enzymatic preparation or protein preparation in the industrial scale and what are their usage. I will now move on to some of the functional non-catalytic proteins. So, they are not catalytic proteins, they are not doing any enzymatic function, but they are very essential or functional. So, functional non-catalytic proteins are those which have properties such as emulsification, gelation, water binding, whipping and foaming, etc. So, not only are the proteins responsible or important for therapeutic purposes, industrial enzymatic purposes or other purposes which have enzymatic function, many proteins are there which do not have any enzymatic function, which do not have antibody generation function, which are not antibodies by themselves, which are not fragments, which are not growth factors, However, they are important in so many different purposes, those are called the functional non-catalytic proteins. For example, kappa casein. Now, casein is a milk protein. Kappa casein is a component of casein which is involved in micelle stabilization of milk proteins and keep the protein suspended uniformly in milk because it behaves like a lipid molecule. This milk 2/3 of the protein is hydrophobic, and the food industry has exploited these non-catalytic proteins. Some of those are written here. So, what are the functional properties of those non-catalytic proteins? It may be used for whipping and foaming, for emulsification, for gelation, for viscosity, water binding, solubility, browning, and even flavor and aroma. And many of those proteins, the mode of action is also given in this table. For example, whipping and foaming, it forms a stable film, and the foods in which this is used, you see you definitely use eggless cakes, desserts, whipped topping, etc. So, in those cases you need to have this kappa casein. You need to do emulsification, which is formation and stabilization of fat emulsions. So, vegetarian sausages, salad dressing, coffee whitener, everywhere you see, if you look at the food systems in which, so baked goods, food, biscuits, confectionery, sausages, everywhere you need some or the other protein to be working. So, these proteins are not essentially catalytic, they are not essentially growth promoting, but they do some or the other function. For example, protein solvation, protein stabilization, protein emulsification, or various other such whipping, foaming, or various other such activities. Another kind of proteins which are of much industrial or other usage is the nutraceutical proteins. So, now this is a new definition which has come through in last 10 years. Nutraceutical is a word which is coined from a combination of nutrition and pharmaceutical. So, it has both the values, that is, it not only protects or gives protection from various infections, diseases by providing a pharmaceutical application of value, but it also gives the nutritional value because of which you get those pharmaceutical applications. So, several nutritional proteins have therapeutic functions. For example, lactose free milk. Now, there are babies who are born with lactose intolerance. So, those people cannot be fed with the normal milk, and they have to fed with a lactose free milk. So, infant food formulations with lactose free milk are now available. Another very important nutraceutical component is whey. What is whey? Whey is the liquid part of curd. You have seen curd. So, if you take out the curd simply by filtering, then you get the liquid. That liquid part of curd is called whey. It is used for treatment from long back, from 16,000 onwards years onwards. So, for the treatment of numerous ailments, for example, jaundice, infected skin lesions, genital urinary tract infections, etc. So, this whey proteins are whey is as simple used in these for these purposes. The solid part, which is the curd, is also beneficial for various intestinal infection. It is used as probiotic, and nowadays this probiotic has become a very important word because it is administered with many antibiotics. So, what happens is when you go to a doctor for some simple ailments, a bacterial ailment, the doctor gives you antibiotic, and if you take those antibiotics, those are many a times broad spectrum antibiotic, and therefore, those antibiotics will kill the bacteria that are present, the good bacteria, the gut bacteria present in your gut, and therefore, it might cause various kinds of problems. So, if you eat curd, it is now considered as a probiotic which is administered most often with the antibiotic that you take, so that this protects or gives a good source for beneficial bacteria, so that the beneficial bacteria can keep colonizing the intestinal tract, and therefore, curd is many a times administered along with antibiotics. So, if you look at the components of milk, these are some of the components. So, per 100 ml of milk you have the protein which is about 3.8 grams, the casein, lactalbumin, lactoglobulin, fat, lactose, the calorific value, calcium, phosphorus, chloride, all of these are having nutritional value apart from the pharmaceutical applications or the values that we just now gave. So, if there is a difference between the buffalo, human and cow, and you can see the buffalo milk has certain components. So, for example, the fat here is about 7.5, grams, whereas the cow and the human has only 3.7 or 3.8 grams. However, rest of the components are more or less the same. So, it depends upon what kind of calorific value you require to ingest, so that you remain healthy. Now, if you look at the components of hay, which I was just now mentioning, and I was talking great about this whey proteins are very useful, and they have really good pharmaceutical and nutrition value. So, the components consists of alpha lactalbumin, alpha lactoglobulin. So, these are two different proteins, and then it has bovine serum albumin. If the milk is produced from bovine, then it will contain the bovine serum albumin. If it is produced from human, then human serum albumin. Then it has immunoglobulins. Look at that, immunoglobulins actually are antibodies. So, it actually gives you certain antibodies and protects you from various infection. Then you have lactoferrin, lactoperoxidase, protease, free amino acids. These free amino acids are also very important. You need to actually have many of these amino acids available to you for your own proteins to be synthesized inside the cell. It has urea, it has growth factors, which helps helps in various kinds of growth related essential components, and they consist of these essential important elements required for health. So, whey proteins results in elevation of a tripeptide. So, if you take whey, then not only does it give all these kinds of components, it actually elevates the proportion of a tripeptide, that means 3 amino acids, and it is a 3 amino acid peptide which is called glutathione. So, it increases the concentration of glutathione in the cells. If it increases the concentration of glutathione in the cells, this peptide acts as a reducing agent and has a broad range of functions. Some of the functions, 2 of those are, so detoxification of xenobiotics. So, if you take xenobiotics, then those xenobiotics can be detoxified. Detoxification of xenobiotics happens because of glutathione, and it also protects the cellular components from oxygen intermediates and free radicals. So, many a times, often because of interaction with oxygen or various other oxidizing agents, we end up generating free radicals in our body. Those free radicals are very dangerous, and therefore, those need to be reduced immediately. Increased levels of glutathione actually helps in reducing those oxygen intermediates and free radicals, and thereby protecting our body from various oxidative damages. Now that we know the components of different protein products, for example, the whey, which has so much more of nutritional value, as well as the pharmaceutical value. So, it helps protection from various diseases, etc. Now, we will proceed to looking at something called protein engineering. So, what is protein engineering? That means protein engineering means you change the protein in such a fashion so that it becomes more useful to you. So, protein engineering is done for the purpose of increasing the stability of the protein. For example, you can increase the stability of a protein against, say, temperature, against different solvents, against acid, and also for plant proteins you can also increase their their stability against environmental stresses like salinity, drought, cold, etc. Many a times we need to increase the activity of the protein. So, for that we can actually again genetically engineer this protein, tweak the protein's active site components, and make the protein much more active. I will show you some examples in due course. And then we can increase the functionality of a protein. So, one protein which is of a particular function or giving some stability or giving some particular enzymatic function, you can actually make chimeric protein by these genetic engineering processes, where two activities of a protein can be combined together in one protein structure, and it will be a bifunctional protein. So, such kind of things are nowadays possible because we know a lot about the processes by which we can manipulate the protein at the genetic level. So, in order to stabilize the favorite protein, however, it is necessary to know the structure of the protein and the components of the structure which are responsible for holding the protein in the shape that it has been held. So, for example, I told you that there are all these non-covalent interactions like the hydrophobic interactions, the interaction between amino acids by van der Waals interactions, hydrogen bond formation or ionic interactions. So, all these interactions together actually constitute the structure of the protein, and because of the structure of the protein or the typical position of the amino acids in the 3-dimensional space, this protein works as it works. However, if you want to tweak or change anything, then what you need to do is you need to actually understand what are these forces, what are the interactions that are happening at these places. Now, some typical examples I have taken here. For example, improving the thermal stability, thermal and pH stability of a protein called subtilisin. Now, this subtilisin is a protease which is obtained from the Bacillus subtilis. This is an organism. Now, this is used in laundry and detergent industry extensively, and because the detergent industry is so big, it is used in huge quantities, in kilograms. Now, there is however a problem. The problem is, if you look at the structure of this subtilisin, it is a typical serine protease, and if you remember my previous class, this serine protease actually consists of these catalytic triad, which is the serine at 221 position, the histidine as the 64 position, and aspartic acid as at 32 position. So, these three which are shown in the blue here actually works on the substrate which is shown in red here, and then this catalysis is done. So, this is a typical serine protease activity. However, as I said, it is used in detergent, laundry. So, it gets easily it gets exposed to bleach, and once that happens, it loses about 90% of its activity, which is a wasteful process. So, we need to somehow increase the stability against bleach, stability of this protein against bleach. So, what people have found out is that just near this serine, very typical, very important serine 221 position, the 222 position is a methionine, which is shown here. Now, this methionine, why this activity goes down? Now, people have found out that this methionine undergoes oxidation doubly, singly or doubly. So, it can form methionine sulfoxide. So, methionine gets oxidized, or it can get doubly oxidized methionine sulfone, and if this happens, then this area lacks the capability to hold on to the substrate, and therefore, this protein becomes inactive. So, what people have done is, after knowing all this information, getting all this information, they changed this amino acid called the methionine 222 without causing any change in the histidine, serine and aspartic acid catalytic triad. They changed this methionine to alanine, and once you change this to alanine, then this protein becomes stable, because this alanine cannot be oxidized by the process by bleach. So, that is a very successful story where the subtilisin has been improved for its activity. I will give you one more example, that of L-asparaginase. L-asparaginase is an enzyme which is used for treatment of leukemia. Now, this asparaginase is generally obtained from E. coli and is slightly less stable. And once we looked at, and this work has been done in our laboratory here at IIT Delhi. So, once we looked at the catalytic residues or catalytic amino acids, we found that there is a typical K represents for lysine, a positively charged amino acid at the 274th position, which was forming intimate contact with another amino acid here. Now, once we knew that, we changed this amino acid to E represents glutamic acid, which is a negatively charged amino acid. So, this is done by genetic engineering, and immediately this opens up a vista of space where the substrate could enter, and it was becoming much more active and stable. So, this activity and stability enhancement was done by simple engineering of the protein. So, there are many such examples, and we will learn about them in different classes. Now, not only are the protein engineering aspects used for changing the stability or activity of the enzyme, we can actually develop new and novel proteins. We have those tools now available to us by genetic engineering approaches. So, we can actually develop immunity development proteins by having some portion of the clones taken from the organisms. For example, if a bacteria infects, the antigenic portion of the bacteria can be cloned into another bacteria or a plasmid, and then specific antibodies against that portion of the antigen can be developed. So, we can have designer antibodies. a novel synthetic gene has been assembled as a first step towards development of a subunit vaccine against hepatitis B virus. So, people have been using the hepatitis B virus surface antigens for development of vaccines, but now we need not have to take that gene, but we can actually synthesize portions of the gene from hepatitis B virus and then get a novel synthetic vaccine that has been developed. Now, we can actually similarly we can improve the nutritional values of cereals and legumes. So, the cereal seeds and legume seeds that have all the proteins, we can improve them. So, whey for example, has the maximum concentration of all the essential proteins, and in these seeds we have the branched chain amino acids, for example also called BCCA, for example isoleucine, leucine, valine and tryptophan, which the structures are shown here. So, you can see in each one of these there is this branching of the chain, it is not a straight chain. These are very important for muscles. So, you can actually increase the nutritional content of these cereals or legumes with incorporation of these particular amino acids. So, that can be actually done by genetic engineering, and then the legumes or the cereals that you take will consist not only of its own proteins, but proteins having branched chain amino acids, and consumption of those branched chain amino acid containing proteins will help you develop your muscles and keep your muscles healthy. Now, whatever you do, finally you need to have some parameter to define the protein value. Though there are two parameters by which we define the protein value as protein engineers or protein experts. Number one is called the biological value and the number two is called the protein efficiency ratio. What is a biological value? It measures the amount of protein nitrogen that is retained by the body from a given amount of protein nitrogen that has been consumed. So, if you have a protein, you define, because you have the structure of the protein, you know the amino acids of the protein, you can know how much nitrogen is present there. So, if you consume let us say 1 gram of that protein nitrogen, and then after some time you measure the amount of protein nitrogen that has been retained by the body. So, that ratio, that is the amount retained by the amount consumed, gives you what is called as biological value. That means, if the retained is less than what is consumed, then obviously the ratio is less than 1, and then the biological value is lesser and lesser if the proportion is lesser and lesser than 1. It has been observed that the biological value of whey proteins, I am emphasizing again and again this whey is very important, because the biological value of whey proteins is the highest compared to even rice, wheat, soybean, egg protein or any other animal proteins. So, all these proteins have biological values, but the biological value which represents the amount of protein nitrogenous content that is retained by the body. So, all these proteins that you take are most of the components of these proteins are going to be wasted, they will not be retained by the body, but the whey proteins have the highest biological value and those are retained by the body. Other one, the protein value index, is protein efficiency ratio or PER. It is used as a measure of growth expressed in terms of weight gain of an adult by consuming 1 gram of food protein. So, if you take 1 gram of protein, how much weight gain you have, that is the ratio, that is the efficiency ratio. Now, how efficient is the protein in gaining weight gaining of an adult? That is what is called the protein efficiency ratio. And again, the protein efficiency ratio of the proteins which are arranged in this fashion. So, this is decreasing order. Whey has the highest protein efficiency ratio. So, whey is very important. What it means is, if you take 1 gram of whey protein, it will be retained and it will result in weight gain equivalently. Milk is the next one. Casein, soya, rice, wheat, etc. As this is decreasing order. So, whey has the highest PER value, milk, casein, soya, rice and wheat respectively. So, with that you now know what these proteins are, what these proteins do, what these proteins can do, what are the ways that we can actually understand these proteins and use those proteins for various purposes. We also now know how to genetically engineer these proteins. We also now know how to produce these proteins in larger quantities. We also now know how these proteins are going to help us attain not only good health, but also good nutrition, and it can be used industrially for various purposes, which does not get limited only to the enzymatic functions or health functions. It actually gives various other functions, and is therefore a very important chapter, very important to be considered in our life. So, with that, I have given you all the information that was supposed to be within the context of this particular chapter, and thank you very much once again for being attentive and listening to me. I would like to again acknowledge all of those for the entire session of these lecture series, and this particular acknowledgement is for those who have contributed or from which I have taken these slides or some diagrams and pictures. I am again re-emphasizing that none of these have any intention to go for any kind of infringement of copyright violation or copyright violations. These I acknowledge sincerely to the contributors, because these have been utilized for only academic and teaching purposes. I thank you once again